Thank you all for joining our last member spotlight for 2020. This series was brought to you by the ASBMR Diversity, Equity, and Inclusion Committee and occurs bi-weekly on Thursdays at 3 p.m. Eastern Time. The series aims to highlight the research of ASBMR members within a collaborative setting and aims in particular to shine a light on the research being done by ASBMR members belonging to groups historically underrepresented in biomedical research. Today's spotlight will have two presenters. First, we have Rachana Vaidya, Ph.D. candidate at the University of Massachusetts Dartmouth. Rachana's research aims to help in identification of key bone-specific biomarkers as therapeutic targets to improve the bone quality of diabetic patients with a higher fracture risk. Our second presenter is Professor Gustavo Duque, MD, PhD from the University of Melbourne, and he will present on a practical approach to osteosarcopenia in a clinical practice and evidence of the falls and fractures clinic as the most cost-effective model of care for osteosarcopenia. As a quick reminder to our attendees, each presentation will take approximately 15 to 20 minutes, and following each presentation, there will be opportunity for question and answer and discussion with the audience. As a reminder, if you are not asking a question or participating in discussion, please keep yourself on mute to minimize any feedback. And yeah, with that, I'd like to invite Rachana to begin her presentation.
All right. Can I share my screen? All right, can everybody hear me? Okay, so our lab works on understanding how the bone quality is affected in diabetes patients. And my thesis focuses on investigating how osteocyte cell function is altered in a diabetic state. So just to give a brief introduction on what is diabetes, it's a metabolic disease in which the body's inability to produce any or enough insulin causes elevated levels of glucose in the blood. Now, there are two types of diabetes. Type 1, in which the body cannot produce insulin, and type 2, where the insulin is used inefficiently. Type 1 develops in childhood, and type 2 is most common in the age group 40 plus. Type 1 is caused due to when the immune system starts attacking the insulin producing cells, whereas type 2, there are many causes and like unknown causes, but it's mostly due to overweight, poor diet, and inactivity. So more than 30 million Americans have diabetes today, and the number is just increasing exponentially. So the high glucose of level, also referred to as hyperglycemia, causes a lot of diabetic complications. It can give rise to stroke, cataract or glaucoma, tooth decay, atherosclerosis, and other cardiovascular diseases. Uncontrolled glucose levels can lead to nerve damage eventually. It also leads to kidney disorders and nephropathy. But however, there is an underappreciated risk of diabetes which has recently gained attention, which is an increased fracture risk in the diabetic patients. So the bone strength is sorry, bone strength is characterized by bone mineral density and bone quality. So type 1 diabetes have a 6 times greater risk of fracture, whereas type 2 diabetes have a 3 times greater risk of fracture. Now, the fracture risk in type 1 diabetes can be explained by their low bone mineral density, and low bone mineral density can help predict the fracture risk in type 1 diabetics. However, the bone mineral density in type 2 diabetes is same or even higher than the non-diabetics. So Uh, we cannot use bone mineral density as a predictive tool to understand fracture risk in diabetic patients. Hence, we got to look at something other than bone mineral density. So we look at bone quality. Bone quality is maintained by the amount of collagen crosslinks or accumulation of advanced glycation and products in the bone. It is also maintained by the bone cells and the tissue turnover, and it is also maintained by the amount of micro damage or the fracture repair that is that happens in the bone. So our focus is on the collagen crosslinks or advanced glycation and products and how they affect bone cell behavior. So advanced glycation end product is a familiar culprit, and it is associated with not just diabetes but a lot of diseases and with normal aging process as well. So, what are AGs? When proteins, lipids, or nucleic acid spontaneously react with sugar in a non enzymatic reaction, they give rise to early glycation products, which then accumulate and undergo condensation and dehydration reactions to become advanced glycation end products. Now, the formation of early glycation end products takes days to week and is a fast and reversible process. However, the formation of AGEs happen over months to years, and it's a slow process. So the spontaneous reaction with the sugar first changes the functional changes, causes functional changes in the enzymatic activity of the proteins, and increases the vulnerability to oxidative stress. Early glycation products exert oxidative damage to the cells and tissues that they accumulate in and give rise to inflammatory reactions within the cells. And AGEs then accumulate in the long-lived proteins such as collagen and from crosslinks and deteriorate the quality of collagen, thus affecting bone quality. So we are not just aging, we are aging as well. So AG accumulation happens in all of us, but diabetic complications or increase the excess glucose in the blood just accelerates the formation and accumulation of AGs in these individuals. And there are two sources of AGs. One is endogenous, where the AGs are formed within different tissues due to the excess glucose in blood. And there are dietary sources, such as foods processed at high temperatures, processed meats, cheese, sausages, foods that are roasted and broiled, which have this really nice color and texture. AGs are known to increase the flavor of the food, but they have a downside and they affect all the tissues they are accumulated in. So, a list of AGs that are associated with diabetic complications and are found to be increased in the blood or serum or urine of the diabetic patients are pentosidine, N-carboxymethyl lysine, methyl glyoxyl, and glucosapine. Pentosidine is a fluorescent AGE, and it is easy to detect and assess in bone as well as serum and urine. N-carboxymethyl lysine is one of the, and methyl glyoxyl is one of the early glycation end products. They are non-cross-linking AGEs that accumulate in the bone, not just bone, but in all tissues. They're recently found to be accumulated in higher amounts than pentosidine in the bone. And glucosapine is a non-cross-linking, non-fluorescent AGE, so it has been difficult to detect it in the serum as well as other tissues. So, what role does AG play in bone? There are two. AGs exert its effect in a physical way and a biological way. Physical effect is by its binding to the collagen and uh, deteriorating the collagen quality by the bone matrix cross-linking. So collagen is known to have enzymatic cross-links, which gives the collagen its strength. However, AGs are non-enzymatic cross-links that form aberrantly in the bone, and they sort of deteriorate the quality of the matrix. This can lead to reduced in mechanical properties such as ultimate strain and stress, or the post-yield properties which are governed by the organic matrix. The biological effect is in terms of altered cellular activity, increased inflammation, which again alters cellular activity, and in turn leads to more accumulation of AGs in the matrix. Together, they also control the fracture healing and increase microdamage accumulation, and thus they have an overall negative effect on the bone and might be playing a role in increasing the fracture risk. So we are going to study the biological effect on bone and how it alters the cellular activity. Now, effect of AGEs on bone cell behavior has been studied earlier, and mainly they have found to increase uh, apoptosis and osteoblast, reduced in differentiation and mineralization in these cells. In osteocytes, they're found to increase apoptosis of these cells, which might play a role in increasing the cortical porosity of the bone. They're also found to increase sclerostin, which is a negative regulator of bone formation, and decrease rankle expression, which like promotes osteoclastogenesis. Thus, it plays an overall role in reducing the formation and reducing the bone remodeling parameters, which ultimately leads to sort of increasing these crosslinks in the matrix that is around the cells, and it forms like a feedback loop around it. So osteocytes are like the key regulators of bone remodeling. They're really great cells because of their location and morphology. One, they're the most 95% of the bone cells are osteocytes. Their location and they are embedded in the mineralized matrix, which means that they must have a greater impact on the matrix around it. They communicate with both osteoblast and osteoclast by secretion of sclerostin and rankle. They are also in touch with the vasculature, so vascular supplies, so they can sense local mechanical strains. They're influenced by the hormones, and they control bone formation and resorption by secreting sclerostin and rank ligand. So they also play a role in repair, such that apoptosis of osteocyte around the microdamage triggers osteoclast and osteoblast to come and like repair the matrix and like induce bone formation in that area. So why are we studying osteocytes? First, they act as endocrine and paracrine cells that regulate the activity of other bone cells and distant organs via secretion of osteocalcin, and thus are key players to study disruption in bone remodeling in diabetes. So these are the major remodeling markers that osteocytes secrete. Sclerostin is a negative inhibitor of the VNT signaling pathway, and thus it inhibits osteoblast bone formation by osteoblast, and it produces rank ligand, which binds to the receptors on osteoclast and helps in their maturation and promote osteoclastogenesis. Now, previous studies have used osteoblast to study osteocyte response for a long time. And it makes sense because osteocytes are osteoblasts which have embedded themselves into the bone matrix. However, the recently developed MLOY cells were also used to study osteocyte response, but they still do not express all the osteocyte markers. So we are studying the response of in osteocyte 454 cell line, which was developed by Dr. Paula Pajevic's lab, and it expresses high level of sclerostin, which is a late osteocyte specific marker. It shows mature osteocytic phenotype, and thus it's appropriate for measuring expression of osteocyte markers, which might give us a true osteocyte response since it has low osteoblast expression markers. So these are some pictures of osteocytes. You can see they have like long dendritic processes and a single nucleus. So our overall goal is to understand how increase in AGEs, hyperglycemia, and inflammation affects the cellular activity. So we think that hyperglycemia leads to increase in AGEs, and AGEs in turn affect inflammation, which might be the cause of disrupting the bone remodeling markers in osteocyte cellular activity, which might help explain the reduced deteriorated bone quality in the diabetic patients. So my hypothesis and specific aims are, um, we think that osteocytes grown in a high AG and high glucose environment will have an increase in expression of inflammatory and glycation markers, which play a contributing role in disrupting the bone remodeling process. So my three aims are: one, to determine how high glucose regulate osteocyte bone remodeling, glycation, anti-glycation, and inflammation markers. Then to check how AGEs such as pentosodine regulate the same markers, and then to determine whether if metformin can improve the bone remodeling parameters via its anti-glycation and anti-inflammatory activity. So the mechanism of action behind this is that advanced glycation end products bind to two kinds of receptors in the cells. One is the receptor for AGE's RAGE, and one another is anti-glycation receptor AGER1. Now, inducing RAGE leads to increase in nuclear fat NFK beta, and which then translocates to the nucleus and increases the expression of inflammation and oxidative stress markers in the bone. Whereas AGR1 is an anti-glycation receptor, and when the AGs bind to this, it sort of has an inhibitory action on the nuclear factor kappa beta, and it leads to endogenous degradation of these AGs. So they help in clearing out the AGs and sort of detoxifying the AGs in the cells. So first, we want to see if we want to differentiate the AG and hyperglycemic response of the cell to then understand, and then to understand how the glycation markers can explain changes in bone remodeling markers and inflammatory markers in osteocytes, then to investigate whether metformin can have an inhibitory effect on the RAGE signaling and inflammation, thus improving the bone remodeling markers in osteocytes. So the methodology that I'm going to use is we're going to grow these osteocytes in media containing high glucose, hyperglycemic media. I've chosen 22 millimolar glucose concentration, which matches like the late diabetic stage profile. Then we are going to check we are going to grow these cells in pentosodine media. I'm going to use two concentration of pentosodine to check if it has a dose dependent response in it or not. And we are going to check if the metformin, adding metformin to to this media has an positive effect on the cells or not. So we incubate the cells at 37 degrees Celsius for 14 days. Since the sclerostin markers are expressed at its peak after 14 days of incubation, and then look for gene expression markers by PCR and protein expression markers by ELISA. So I'm going to look at three different types of marker: bone remodeling marker, which includes sclerostin, rank ligand, OPG, and cathepsin K. Cathepsin K plays a role in perilacunar remodeling response. So I also want to check if it has an effect on the perilacunar modeling through osteocytes or not. Then we want to look at how the inflammation or pro-inflammatory cytokines response is changed in response to high glucose and pentosodine. And we want to check the response of these cells of hyperglycemia and pentosodine on the RAGE and AGR1 markers. So we've done some pilot study data on glucose incubation, and so we grow the cells in control media, which has 5.56 molar of glucose concentration, and then a high glucose of 14 millimolar and 22 millimolar. We found that sclerostin expression was increased threefold and sixfold in high glucose concentration. The same with however, there was no difference in rank ligand expression in these cells. We also checked for the protein expression in these cells in the cell supernatant, and found that sclerostin was increased fourfold and fivefold in high glucose media, whereas the rank ligand showed no change in expression in the control versus 22 millimolar, but there was a sudden rise in peak. There was a peak high increased expression in 14 millimolar glucose. But this was done from one study, so we don't know whether something went wrong with the study or this is like a there was no significant increase in any of the groups. So We then wanted to check if we can grow the cells on glycated collagen. So for that, we took rat tail collagen and we glycated it with methyl glyoxyl. And then we coated the cells, the plates with these glycated collagen and grew the cells on top of this coating. So when we checked the expression of sclerostin in control versus glycated collagen, there seems to be a trend of decrease in sclerostin expression, but there was no significant change in the day 1 versus day 12 of cells grown in control versus glycated environment. We also check the effect of expression on the protein markers, and there was no change found in sclerostin expression over the day 1 and day 5. Although a little higher, there was no significant change in it. We also found the change in rank ligand expression, and you can see the control, there's no change in the control versus there's little higher rank ligand expression in glycated, but there's no significant changes. Also, the same for rank protein. The protein data look very different than the gene expression data. There seems to be a decrease in the rank ligand expression of soluble rank ligand protein markers, but there's no significant changes in any of these groups. So we thought that maybe since these osteocytes are embedded into the bone matrix and they are in a 3D environment with the glycated, with the collagen or the collagen glycation, it might be better to study that in a 3D cell culture versus a 2D cell culture. So we sort of decided to drop this and move ahead with the 3D culture. There's a lot of work that I need to do, but because of this pandemic, everything has been delayed and a lot of things are not working. So currently, I'm testing new primers that I've got and optimizing the PCR condition before I do my experimental runs. So I've grown my cells in high glucose and pentosirine media, but I've not been able to get any data out of it yet because my PCR primer efficiencies are a little weird for some of the primers, and I'm trying to get that within the 90 to 110 range before I do any experiments. Then we were testing some new ELISA kits, and we found a few bad batches of ELISA kits, which has made my which for which I don't have any protein readings as well. And again, now we are culturing the cells in pentosirine media. So I use the synthetic pentosirine that we get from Cayman Chemicals and add that to the aqueous media. But I need to test if the pentosirine stays stable in this aqueous media for at least 14 to 30 days, for which, because that's how long we'll be growing the cells in, for 14 days and maybe much later than that. So I'm using HPLC to test if the to check if the pentosirine in media is present and stays stable for 14 to 30 days. Now, I'm also deciding how to use metformin. There are two ways I can do it. One is to treat the cells for 14 days with glucose and then add metformin for 24 hours to check the effect. And or I'm thinking whether I should add it with the high glucose and pentosirine media for 14 days to see if there is an inhibition, because when metformin is known to interact with the dicarbonyl groups, so maybe it interacts with the pentosirine and doesn't allow them to enter the cells or fights for it, an inhibitory, like it inhibits the process before it enters the cells. So that's what I'm doing. Significance of our research, we think that our understanding the role of AGEs and osteocytes will allow for use of anti-glycation agents in diabetic therapy, use of osteocytes as target cells in diabetes treatment. It could lead to use of pentosirine as a biomarker for deteriorated bone quality in diabetes. And using AGs as therapeutic targets is not only benefited in diabetes, but many other chronic diseases involving AGs, such as Alzheimer's and cardiovascular diseases. It will also help in promoting the research and the identification of other AGs in bone, because since currently we know that only pentosirine is in high amount, CML is recently found to be in high amounts, but there are many other AGs which have not been identified in bone yet. And ultimately, this should lead to development of assays or diagnostic tools that can measure the glycation of proteins to indicate bone health in patients. So the overall goal of our project is that based on previous findings, we suppose that AGEs might be involved in increasing the fracture risk by deteriorating bone quality, independent of the bone mineral density, by acting directly upon osteocytes. Unfortunately, little is known about the role of AGEs in osteocytes. So this study aims to determine whether AGEs, or more specifically pentosidine, which is found to be in high concentrations in bone, can, in a mature osteocytic cell line like OCY454 cells, induce apoptosis, increase production of inflammatory cytokines, and alter bone remodeling by increasing or reducing the turnover. And this will help in understanding one of the few mechanisms on how AGs play a role in deteriorating bone quality in diabetic patients in order to target AGs as a part of diabetic therapy in future. I'd like to thank my committee members, especially my advisor, Dr. Lamia Karim, to invite me to this talk. I was really scared to present in front of everybody, but thanks for making me do this. Also like to thank my lab members with whom I work every day. Tarane, she's another PhD student in our lab. Lauren and Morgan are like master students, and our funding sources, NIH, ASBMR, and the cell source and training support, which was given by Massachusetts General Hospital and Centers for Skeletal Research. So thank you, and I'll take any questions now.
Can I ask a quick question about why you chose to use metformin? Is metformin known to have a direct effect on the osteocyte? It's very different when you use it in cell culture versus in the whole organism.
Yes. So metformin is known to target the MAP kinase pathways in the cells. So we initially thought that what if, so how people, like how other researchers are using vitamin B and aminoguanidine to study their effect in like tissue. But I don't know if it has a direct effect on tissue. So it would be better to study it in cells. So and metformin is known to have anti-glycation and uh, re- uh, anti-inflammatory effect. So it would be better studied in cells.
But
Okay.
isn't that effect in the whole organism? Because when you give someone metformin, obviously there's a bunch of things that go on. Maybe
Yeah, I mean, yes. So there are studies which have. So there was a recent study by in Japanese patients where they gave metformin for 6 months and checked if the level of serum pentosidine levels dropped in these patients. And after 6 months, they found that serum pentosidine levels were lower in these patients compared to the control group. So how is metformin does metformin has beneficial effects on the bone health of people as well, can be probably studied in cells. So I thought that it would be great to check its effect on the cells.
So yeah, can I ask a question? Your goal is to look at AGE effect on osteocytes. So you propose all these in vitro studies. Have you think about any in vivo studies? Because
So, I mean, it would have been great to study it in vivo, but in our college, we don't have access to animal models and we don't have an animal facility to study in vivo models. So I thought of starting by in vitro cells, and then with the data, we can probably propose its study in vivo models.
I have a question also. Um, Instead of looking to see the effects of AGEs on osteocytes, have you thought about looking to see if osteocytes can cross-link the collagen and create AGEs, especially in response to high glucose?
That's a good question. So one way of studying this is to culture these cells in a 3D gel in which in which you have collagen, and then you put these cells in high glucose media and then study if it has an effect on the collagen around it or not. So I do plan to do that, but I thought it would be better to first check how these AGEs and hyperglycemia affects the cells and then study it in a 3D system. But yes, that is one of my aims too.
Rachana, wonderful talk. I'm going to throw back more towards the immune lineages and ask, have you thought about taking osteoclasts or myeloid cells and different, look how they behave with the collagen that's with the different glycation products and see if it changes the myeloid signaling and some of those inflammatory reactions that could be happening with osteoclasts?
So there are a lot of studies which have already checked its effect on osteoblast and osteoclast. And yes, it does affect the way the cells differentiate. And that has been studied yet. But since osteocytes are like embedded in the matrix, I want to see how osteocyte behavior is changed, because if we can control or manage the or inhibit these AGs and osteocytes, they could have an effect on the osteoclast and osteoblast as well, since they are like the key regulators of bone remodeling.
Nice presentation, Rachana. So I had a question. Can you talk a little bit about your transition from the 2D cell culture to the 3D and why you think it might make a difference in some of your outcomes?
So these cells have long processes which are extended into the embedded matrix. And when you study it in 2D, I don't think that response is like truly, what do you say? I mean, replicated in a 2D culture, versus when you put them in a 3D culture, they have their true, the osteocytes are in their true self. So embedded within the matrix. So that might be a better model to study the cells osteocytes versus in a 2D culture.
Nice talk. I have a couple of methodological questions. You treated your cells with glucose.
Yeah.
What did you use as control?
So we use 22 millimolar mannitol as control to see if there is any osmotic differences in the cell. I had a hard time growing the cells in glucose initially because the pH of the cells would, the culture media would drop. And even after putting 25 millimolar heat piece, it still used to drop. So rather than growing the cells for 14 days in high glucose media, I thought it's better to grow these cells for 7 days. So I grow for the first 7 days, I grow them in the normal media. And then for the next 7 days, when I get the peak sclerostin expression on the 14th day. So I have to grow them for 14 days anyway. So I grow for the last 7 days, I grow them in high glucose media and then check it. So it's been good so far.
Okay, thanks. The other question I have is, we have been criticized because by using glucose, because that's not the only thing that is increased in the serum of the patients with diabetes. And I would argue that the same thing happened with the pentosidine. If you're using isolated pentosidine, you are not reproducing what is happening in the microenvironment that the osteocytes are exposed to in vivo. So I was wondering if you ever thought about using serum from diabetic patients and compare that with the normal serum, or patients with uncontrolled high glucose versus controlled glucose, even with diabetes, and that could make a difference.
A great idea to check serum, but so what I thought is, first we need to see how the pentosidine affects the cells, and then to see if we have serum with pentosidine, because it has many other parameters, how then that together affects the cells. So here I'm trying to just see that actually the aim of this is to see if hyperglycemia has the similar effect as the pentosidine in the cells, especially on the RAGE and anti-glycation markers. Or is it that hyperglycemia paves way for the AGs, so it increases rate signaling, and then the AGs gets accumulated more or not? So that's what I'm trying to do here. But yes, I think further studies should be done by collecting serum from diabetic patients. I think that would be a better study model.
Thank you.
Thank you so much, Rachana. That was a great presentation. And in the interest of time, I'd like to move on to our second presentation. I'm sure if anyone has any other follow-up questions for Rachana, she'd be happy to touch base after the meeting. But at this time, Dr. Duque, I'd love to invite you to begin your presentation.
Okay, so I hope that everyone can hear me and see my slides.
Yes.
Good afternoon, everyone. Good morning from Australia. It's a real pleasure to present today and thank the Diversity, Equity and Inclusion Committee for inviting me today. What I'm going to do is I'm going to share a little bit of the experience that we have here in Australia in my team regarding osteosarcopenia as a new concept for some people. But before that, I just will share a little bit of my life, of my personal experience, by sharing with you a little bit of where I'm from. I come from Colombia, from South America. I was born in a very small town for 120 inhabitants. And then I have moved around a little bit in the world, and I ended up here in Australia after being in North America. And throughout all this career, I have been very positively influenced by the ASBMR, which has facilitated so much of my collaborations and a lot of the evidence that is generated and shown at this, in this talk. So for many years, the concept of osteopenia, osteoporosis, and fractures was a little bit fragmented from what we understood as sarcopenia unfolds. Different groups, different specialties approaching these two entities from separate perspectives. But very much more recently, people started to realize that they are in fact very much connected, and that falls and fractures could share multiple mechanisms, and that in fact muscle and bone were extremely connected, including the way they age. And this is evidence from the Geelong osteoporosis study, which in a collaboration with Julie Pascoe, where we compare the bone and muscle aging in men and women, and you can see the trajectories that they are not identical, but they go more or less in parallel, with a loss of about 0.5% of mass per year after the fourth decade of life. What is interesting is that some people, about 40% actually of our older adults, have a higher and above a loss that goes above what is shown here in these curves. And of course, they become osteoporotic or they become sarcopenic, but there is a proportion of them that have show both entities, both show osteoporosis and show sarcopenia at the same time. And what we have been working in the last probably 10 years is trying to identify these similarities and these shared circumstances that determine that a person loses at the same time a significant amount of their muscle mass and their bone mass. One of the interesting things that we have found is that the risk factors between sarcopenia and osteoporosis are almost identical. And also interesting evidence provided by ours and by many groups around the world is that there is a very active communication between muscle and bone through myokines that could go through the circulation and reach the bone, and at the same time osteokines that could be secreted and produced at the bone by the bone and reach the muscle, regulating the function and growth. So I will not go into all these factors today because of time, but basically what I want to highlight here is that I'm not presenting or talking about a new disease. I'm talking here about a syndrome known as osteosarcopenia that combines two entities, two diseases, osteopenia, osteoporosis, as we understand it very well, and sarcopenia independently of the definition. And I know that Peggy Cawthon was presenting here at these same seminars a couple of weeks ago, and she probably mentioned about the different definitions that we have at this moment and for sarcopenia, but independently of what definition we use, and you combine it with osteoporosis, we are going to identify somebody as osteosarcopenic. And the concept is growing. We can see that more and more people are writing about it. More people are adopting this as a topic of interest. We have focused on different aspects, and these are the aspects that I would like to mention today in case we want to collaborate on different areas. And there is definitely a major need to understand the interactions and the two-way interactions and crosstalks, as well as the mechanism, because we know that there are intrinsic mechanisms, common mechanisms that occur both in muscle and bone at the same time. We know that there is another set of mechanisms that relate with the defective muscle leading to defective bone. And as I mentioned before, mechanisms that explain or that associate osteosarcopenia with defective bone leading to defective muscle. In our group, regarding the pathophysiology assessment of or identification of osteosarcopenia, we are working with the hypothesis that osteosarcopenia is in fact a case of geroscience. For those of you who are not familiar with the concept of geroscience, basically is the explanation for multiple diseases that are associated with aging could be through a very a set of shared mechanisms. And here are what we call the pillars of aging. We are looking at each one of these components, and I will share with you a couple of studies that we have done, or a couple of experiments we have done, or a couple of the evidence that we have seen in, for instance, genetics or stem cells, which is something that could explain the common mechanism that we observe in osteosarcopenia. If we look at the precursors of the muscle and bone cells, well, we have these, of course, the mesenchymal stem cells, which at the same time are precursors or can differentiate into adipocytes. And this has led us to propose that this fat infiltration that we observe in both muscle and bone could be an important pathophysiological mechanism of osteosarcopenia. If you observe a sarcopenic muscle, you will see that there is fat not only between the fibers, but there is also there is fat infiltration inside the fiber, which is interesting. And when you compare a sarcopenic muscle with an osteoporotic bone, you can see that with, of course, evident differences between tissues, you still see that there is a very significant amount of fat infiltration within the bone marrow that mimics also what we observe in the sarcopenic muscle. And a summary of that was our proposed concept that osteosarcopenia is a lipotoxic disease where this fat that infiltrates the muscle and bone has a negative impact and on bone function, bone cell differentiation, and facilitating bone resorption, lipoapoptosis. We have also reported the effect of this fat on autophagy. But what is very interesting is when we compare what we observe in bone with what other people have observed in muscle, the effect of the presence of this fat on muscle and bone cells, both in vitro and in vivo, seem to be very similar. Again, with the different circumstances, with the taking into consideration that bone is a mineralized tissue, but in both tissues, what we observe is a very much catabolic stage induced by this fat infiltration. When we look, for instance, at genetics, which some people say, okay, well, maybe the major difference between osteoporosis and sarcopenia would be from the genetic perspective. A phenogram done by David Karadzic and Fernando Rivadeneira has already demonstrated that there are many genes that have an important regulatory role on bone and muscle mass. I will not go into all of this, but I can just share with you one that is very interesting that we reported with that group not long ago in 2017, where we reported the first meta-analysis demonstrating genetic factors with pleiotropic effects on both bone mineral density and lean mass. But the reason why I'm sharing this with you is because this gene is particularly interesting because it's mostly a regulator of fat, more than a regulator of bone and muscle. Regarding the biology role. Talking about a little bit about the clinical phenotype, we described the phenotype in our cohort in Sydney. This is some from our Falls and Fractures Clinic in Sydney. We found that the people who have osteosarcopenia are usually female, older, and of course all the variables, including bone mineral density and muscle mass and muscle function were affected. But the question at this moment that we are trying to answer is whether osteosarcopenia is a continuum. So where does it start? Does it start with sarcopenia, where we have weakness and falls, and then we develop osteopenia and then osteoporosis, and when they are combined, we have this functional impairment that predisposes to falls and fractures. Then we develop frailty, and then we develop disability. Is this a continuum? And the $5 million question is what starts the process, one, and the second one is where the process starts. It starts as a sarcopenic or osteopenic. This is something we're trying to answer. Or is osteosarcopenia a dynamic syndrome where we have a robust individual with normal aging in their muscle and bone, who suddenly or progressively develops this osteosarcopenia, suffers falls and fractures with all the consequences that we know come from these adverse outcomes, including fear of falling, physical dependence, malnutrition, and poor quality of life. And then one group of them end up quickly becoming frail, disabled, or end up in a nursing home, whereas another group will get again into this vicious cycle. But most of the individuals will probably end up, after having osteosarcopenia, falls and fractures, they will most likely end up as frail and disabled or institutionalized. So the other question is, again, if it's not linear, it's probably a dynamic model, and what can we break this vicious cycle that we observe here? And the other question was whether it's just, we're talking about something that only combines osteoporosis and sarcopenia, or is something that combines everything, osteopenia, osteoporosis, and sarcopenia. And this is something that we reported not long ago, in which we also found that people with osteopenia and have associated with sarcopenia, they also are very much high risk of falls and fractures as compared with the ones who have osteoporosis. So the component of sarcopenia was one of the determinants of this high risk of falls and fractures in osteopenic patients. The other interesting part that I wanted to share with you is regarding the diagnosis of osteosarcopenia. This is a little bit challenging, again, because the multiple definitions of sarcopenia. So we have been proposing that maybe we should look for innovative ways of measuring muscle mass, either by looking at the areas using DEXA, just to look, and with DEXA, as you know, that has the limitations of being measurement of lead mass. But what about if we measure it in specific regions of interest where we have a lot of muscle, and we also have some idea of what the bone mass is? And we did this, we reported preliminary data in this in a cohort of Singaporean individuals, older individuals, male. We found that the sensitivity to diagnose osteosarcopenia from this method was almost identical to using the other methods. And coming back to the story about the fat infiltration, we are developing some methods to identify and quantify fat. And amongst those methods, we have developed a software that is called Tissue Compass, which is able to, has the capacity to quantify fat infiltration and also bone and muscle tissue in CT scans. This is something that we are at this moment validating with the Mysteros study. But basically, our working hypothesis is that if we can quantify fat, we can predict adverse outcomes, and at the same time, we can diagnose both osteoporosis and sarcopenia just using a few CT scan sections. So the last part of my talk that I wanted to share with you is how are we going to, if we're going to have identify this diagnosis in our patients, what are we going to offer them? And this is where the challenge is, because, and I've shown you here the Holy Grail, because probably the person who discovers an effective treatment for both entities, for osteoporosis and sarcopenia, will identify the Holy Grail in the musculoskeletal and probably in the geriatric field. So there have been some studies looking at pharmacological approach, of course, the non-pharmacological approach, and I will not talk very much about the future directions, but I'm happy to discuss it with you offline or online. The non-pharmacological interventions, the usual ones, and we know that there is evidence that against and pro these interventions, vitamin D, but protein intake, calcium intake. But all these interventions have demonstrated to be effective in terms of keeping our bone and muscle health, probably not always effective on preventing falls or fractures, but they are being always associated with as coadjuvants when we treat somebody for osteoporosis and sarcopenia. So we still recommend this non-pharmacological approach. The challenge at this moment are the pharmacological interventions, and I will give you an example that I find very interesting, which is Denosumab. Everyone here is very familiar with it, and Serge Ferrari did a very interesting study, which included animal studies and also human studies, where he treated humans with Denosumab, and additionally to finding the same that we know about Denosumab regarding bone mineral density, they also found that the Denosumab treated group showed a better appendicular limb mass and better hand grip, suggesting that there was a therapeutic impact of Denosumab not only in bone but muscle. We did a similar study, a very small one, a pilot with our patients at the Falls and Fractures Clinic in Melbourne, and we identified actually a very interesting set of effects, not only from Denosumab, but also from zoledronic acid. By the way, Serge Ferrari didn't find any effect of zoledronic acid. We found a smaller effect, of course, of zoledronic acid, but much wider in terms of all the variables that we measure when we look at muscle function. So still, there is this letter to editor recently in JBMR where we there's definitely agreement that we need more evidence to really suggest that Denosumab has an impact on both. Well, we know that it has an impact on bone, but it has an impact on muscle, and whether the impact is also effective on preventing falls. This is a quick summary of where the pharmacological treatments are. I already mentioned about Denosumab, and the results from testosterone and growth hormone, although they show some improvement in mass, that we know that they are not being very effective on preventing adverse outcomes. And with anti-myostatin antibodies, a very promissory treatment that was expected to increase muscle mass and at the same time probably have an impact on bone. Unfortunately, the most recent clinical trials have shown a lot of or a very strong effect regarding muscle function and also regarding bone markers. Finally, I started talking about the fragmented model. How can we avoid this fragmentation? How can we make that the patient who is at risk of has already osteoporosis and sarcopenia goes to the same unit, goes to the same, and we see that sometimes that those patients have to go to 2 different clinics, 2 different groups. So we proposed the creation of an integrated model of care that involves assessment not only for muscle mass, muscle function, but also bone mineral density, and a complete comprehensive identification of the risk factors for both entities. We, of course, integrate balance and gait assessment, also integrating some new technology. And this is a small figure, but basically what we have done is we evaluated the pre and post 6 months difference between people who attended the clinic, and we found that there was a very significant change in the level of risk for falls and fractures 6 months later after the patients attended the clinic, after following all the recommendations or most of the recommendations that we made. We were also successful in changing risk factors, not only for falls, but also for fractures. So there is definitely a combined benefit of exposing or seeing these patients at an integrated setting. And this is more or less the clinical algorithm that we proposed for osteosarcopenia, where we can identify at the same setting who has osteoporosis, who has the risk for fractures, who has the risk for falls, and who has sarcopenia, who has both of them. And then we can establish with the resources we have in terms of therapies, what the plan of the care plan for these patients will be. With this, I would like to thank our team at the Australian Institute for Musculoskeletal Science, our collaborators all over the world, and of course the people who pay the bill, the Australian NHMRC and the MAC here in Melbourne. And with this, thank you very much, and happy to have a discussion or answer the questions. And if we don't have time, I'm happy to do it offline as well. So please feel free to contact me. Thank you very much for the opportunity.
Hi, Gustavo. I have a question. On one of your earlier slides, you had an image of a muscle cell with, or a muscle with the writing something about peripheral atherosclerosis. Can you explain what you mean by that?
Well, that's, I would say that that's, well, in general, the concept of atherosclerosis, I mean, I was referring to that. The figure was taken from another publication. So this group has just summarized all of them. We have not looked at atherosclerosis in the context of osteosarcopenia yet. But we know that atherosclerosis has been clearly associated not only with low bone mass and with, especially with the increased adipogenesis in the bone marrow, but also it has been associated with poor muscle mass and function. So I would say that will be the explanation for that group to include that concept there specifically.
Okay. Have you considered then, I know that you're kind of making the link between, I guess, fat infiltration and atherosclerosis. Have you considered looking at overall vessel density in the muscle or inside the bone and bone marrow, and making the link between the fat infiltration and the loss of blood vessels?
Yes, this is a good point, and definitely we are trying to do that. We are trying to use this software that we developed to identify also that component. That there are some softwares that already can tell us at the level of irrigation and circulation in the muscle using imaging. So yes, definitely. And there are other dynamic methods to identify or quantify the level of perfusion of the muscle and bone. So definitely that's something that we are at this moment.
So we know in bone there's a loss of blood vessels as well as perfusion with aging.
Exactly.
And in the muscle, it's not so clear. I know there's a loss of perfusion, maybe not so much a loss of density. So would it be safe to say, or would you be comfortable making the assumption that it's actually the loss of vessel that leads to fat infiltration that then leads to osteosarcopenia?
Could be one of the factors, I agree. But I think the dependence on, and you're right, the dependence on circulation is different, because you look at the O2 saturation, local O2 sat in the bone marrow versus the muscle is very different. The bone marrow is extremely low O2 sat, in the muscle is different. So yes, definitely there must be a link, but I don't think it's the only or the most predominant risk factor, I would say.
Okay, thank you.
No worries. Stay quiet. So, so as I said, I'm happy to discuss it offline as well. I mean, feel free to write me an email. I'm happy because we cover from basic sciences into clinical sciences. We have a very much translational group, so I'm happy to discuss any other components offline.
Yes, I was about to say, I know we're getting to the top of the hour, so I do want to be respectful of everyone's time, but I do thank you both for two wonderful presentations to end the year off strong for our Member Spotlight series. I welcome you all to sign up to present or nominate a colleague to present for our 2021 calendar schedule of Member Spotlights, which will be published soon. So please keep an eye out, and we hope to see you in the new year. Bye, everyone.
Thank you, everyone.
Bye. Thank you.
Bye-bye.